Hey everyone. So in the last video, I believe we were talking about RNA transcription, how RNA polymerase, or the various RNA polymerases in eukaryotes transcribe RNA from DNA. In this video, we are going to talk a little bit about RNA processing, which is pretty important in eukaryotes. So just briefly, before we get started, let me discuss an important difference between prokaryotes and eukaryotes with respect to transcription. So prokaryotes over there, eukaryotes over here. Now, if this is a gene, or a piece of DNA of a gene, and here is RNA polymerase, and I'm not showing the transcription bubble. So just a very basic diagram of transcription. And let's say this is the nascent RNA. And just like DNA, RNA has a directionality. So let's put the 5' end here, and the 3' end is up there, and the nucleotides, the ribonucleotides, are being added to the 3' end. So in prokaryotes, the ribosomes can jump on to that messenger RNA as it's being transcribed. And we can actually get multiple ribosomes. And I don't know why they should be the same size. My diagram is not very accurate. And I can't keep making them bigger. I don't mean to do that. But, you know, as the ribosomes progress down the messenger RNA, this is the growing polypeptide chain, we would expect the polypeptide chain to be bigger or longer. Say this one here just started translating, so the peptide chain is relatively short. This one has been translating for a while. Peptide chain is very long. Eventually it will hit the stop codon, and that polypeptide chain will be released. And it will fold into a protein, and with whatever function of the protein from that gene that is being transcribed. So in prokaryotes, transcription and translation, which is the process of reading the messenger RNA to synthesize the protein. These two processes can happen at the same time. So they can happen at the same time in prokaryotes. Now eukaryotes, okay, transcription is generally occurring in the same manner. Here's RNA polymerase. We have either 1, 2, or 3. You know what? Let's go with 2, right? Because we're talking about messenger RNAs here. So RNA polymerase 2. This one, remember, RNA-P, 1 RNA polymerase in prokaryotes, at least 3 in eukaryotes. RNA pol 2 is the one in charge of transcribing protein coding genes. So here's the nascent RNA. And remember, the nascent RNA is the RNA as it's being transcribed. It's a name for an RNA that is in the process of being transcribed. This is also the pre-messenger RNA. Now, when transcription finishes, we have a pre-messenger RNA, no longer a nascent RNA. It's been released from RNA pol 2. So we have the pre-messenger RNA, and it must undergo processing. So it must undergo various processing steps before it can be exported to the cytoplasm where translation occurs. So in eukaryotes, transcription and translation occur at different times. And we can add in different locations. Right? Transcription occurs in the nucleus, translation occurs in the cytoplasm or on the endoplasmic reticulum, which is outside of the nucleus. But in prokaryotes, again, transcription and translation can happen at the same time in the same place, essentially the same place. So today we are mostly going to talk about processing, what the various processing events that the pre-messenger RNA goes through before it is exported to the nucleus for translation in eukaryotes. Okay, so what are the processing events? Now, let me start with diagramming a protein coding gene in eukaryotes. I will title this RNA processing, and I will specify eukaryotes. I'm not really sure of the processing events that prokaryotic messenger RNAs might go under. They might not go under any. Like I said, transcription and translation can occur at the same time. I'm not really sure if anything happens to the 5' end of a prokaryotic messenger RNA. So let me just specify eukaryotes here. And the major concept we will learn essentially is that RNA processing is much more important for eukaryotic messenger RNAs than prokaryotic messenger RNAs, which I know very little about. But I know enough about RNA processing in eukaryotes. So let's talk about that in more detail. So I'm going to diagram this protein coding gene in eukaryotes in a way that I haven't really done before. So I'm going to put these little boxes in here. And you'll see what these boxes are for in a moment. So this is the DNA region. This is the DNA of the gene, diagramming the DNA level. So this is the transcription cleavage site, TCS. This is the transcription start site, also known as the +1 site. This is the promoter region. This is the terminator region. So those are all things we've gone over before. Now, when this is transcribed, it's going to give rise to a pre-messenger RNA that looks something like this. And here's the 5' end. Here's the 3' end. And it's going to undergo at least 3 processing events. So the first one is going to be adding a cap. It's going to be the 7-methyl guanine cap. And it's added to the 5' end of the messenger RNA. Now another processing event is the polyadenine tail. So what is that? So adding 100 to 300 adenine ribonucleotides to the end of the pre-mRNA. And the third processing event we will talk about is exon splicing. Now, so that's what these boxes are. These are the exons. And the lines, these are the introns. And the introns are going to be removed from the pre-messenger RNA. and the exons are going to be joined together. Now, interestingly, introns make up 24% of the human genome. And you might think, by the way the diagram is drawn here, that, okay, a similar amount occurs with respect to exons, but no, that's not true. Only 1% of the human genome is made up of exon coding sequences. So much more, about 24 times more of the genome is intron coding sequences, or it's comprised of intron DNA that encodes introns, and 1% is exons. So a lot more introns. So what are introns? Like, why is so much of our genome made up of this? One would think exons are more important. So we don't really know. We don't really know why introns are, make up so much of our genome. We know they're important, and we're going to show. We're going to learn today why they are important. But we don't really know why sometimes they are so long. So these are the 3 steps we're going to cover. We are not going to cover another one, RNA editing. So there's a process in animals, and now we're learning fungi too, where and I guess all eukaryotes, where you can change the bases in an RNA to a different base, and that can happen after transcription. So that's a type of RNA processing. So there's a common one where you change the cell change adenines to inosines. This is a base we haven't talked about yet. So it's a modified base. It's not a standard base. But when you change an adenine to an inosine, well, it can refer to a different codon. So you can change the code of the messenger RNA by changing adenine to inosine. But we're not going to talk about RNA editing in this part of the course. Okay, so let's talk a little more about these events one at a time. So what is that 7-methyl guanine cap? What does it look like? 7. So 7-methyl guanine. Now let me start by diagramming, say, the first nucleotide, or the first 2 nucleotides of a messenger RNA. So here's the 5-carbon sugar, 1', 2', 3', 4', 5'. Now let me put a phosphate here. Well, I always put that double bond in way too soon. So let me put 1 phosphate group on there, and get a sharper pencil. And let me put, oh, it always breaks, right? Let me diagram the other 5-carbon sugar here. Sometimes I put the carbons in, sometimes I draw them as angles. And I'm going to put just other nucleotides down here. Other nucleotides in the RNA chain. So let's put the bases here. Let's put a G here, and well, let's put an A here. Okay, so we have the first 2 nucleotides of an RNA chain, and these are ribonucleotides, right? So we should have hydroxyl groups attached to the 2' carbons. So the 7-methyl guanine cap, now it's attached to this guy right here, this 5' carbon, and it's attached not through a single phosphate, but 3 phosphate groups. Okay, now let me diagram that 5-carbon sugar. 7-methylguanine is just a nucleotide. It's just a guanine nucleotide, and we'll talk about this 7-methyl in a moment. So here's the 5-carbon sugar. Now, this diagram is wrong, isn't it? Yes, it is. And I'm running out of space, unfortunately. Okay, so here's the 5' carbon here. Now, there is the 4. No, there's the 5, yes, 5, 4, 3, 2, 1. There's the 1' carbon. This is going to be attached to the guanine nucleotide, right? Because the 1' carbon is attached to the guanine nucleotide. These are hydroxyl groups. Remember, the O's are attached to the carbons. Now, not a very great diagram here, but you can kind of see, right? So this is the 5' carbon of the nucleotide. It's attached to a phosphate. This is the 5' carbon of this nucleotide. It's attached to a phosphate. And then we have another phosphate between them. So we have 3 phosphates linking the first nucleotide of the messenger RNA to the 5' 7-methyl guanine cap. So let's say this whole thing right here, we'll call this whole thing the cap. So what is this 7-methyl guanine? This is a guanine nucleotide. Well, it just means there's a methyl group in CH3, that's methyl. The methyl group is attached to the number 7 position on the guanine base, nitrogenous base. And like I said, we don't need to memorize the structures of the nucleotides in DNA and RNA. We did memorize the general ring structures of purines and pyrimidines. So briefly, ring structure, right, of a purine looks like this. And these are the numbers given to the atoms in this ring structure. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is where that methyl group would be. Okay, so that's the 7-methyl guanine cap. This is the 7-methyl guanine nucleotide. It's attached to the first nucleotide of the RNA chain through 3 phosphate groups. So the next one is very easy. So the next processing event we want to talk about is the poly-A tail. Still recording, good. Now, okay, poly-A tail, I'm not even going to diagram it here, anything here. So added to the pre-messenger RNA by a protein called poly-A polymerase. Via a template independent process. So what does that mean? Why do we even bother saying via a template independent process? So remember when we talked about DNA replication and even just RNA transcription, those processes are using a template, and they're adding nucleotides to the growing chain of either DNA or RNA based on the nucleotide present in the template. Here, however, let's say this is the last nucleotide of a pre-mRNA with an OH group at the 3' end, at the 3' carbon. So what this poly-A polymerase does, it just adds A's, right? To the end. No template needed. No template needed. Okay, so that's what we mean by poly-A polymerase. Adds, and I think we can call this PAP, I think that's standard PAP, the protein PAP, adds the adenine ribonucleotides via a template-independent process to the 3' end of a pre-messenger RNA. So that's step 2. Now, okay, let's take a look at splicing now, the third processing event. So splicing, we talked a little bit already about 24% of the human genome being made up of introns and only 1% being made up of exons. We'll talk more about what the other percentages of the human genome have or are made of. Essentially, most of it's repetitive DNA. We'll talk more about repetitive DNA towards the end of the course. So just before we go on, in our recommended textbook, table 13.8, we have an interesting table here. So we are going to talk about, just briefly, lengths of genes. So gene length versus mRNA length versus numbers of introns. So with respect to genes, we're talking about protein coding genes here. Protein coding genes give rise to messenger RNAs, and they also have introns, typically have introns in eukaryotes. So we're also going to use a term called KB, which is kilobase. So a kilobase is just 1,000 bases, because we're going to use some pretty big numbers here. So we're going to want to talk about these numbers in kilobases, so 1,000 bases. So the first gene in the table is the insulin gene. It is 1.7 kb long. The messenger RNA is 0.4 kb long. I'm guessing that doesn't include the poly A tail. And there are 2 introns found in the insulin gene. So it looks like, about 1.3 kb was intron sequences. So that was removed and disposed of while making the the mature RNA. So another one, albumin, which I believe is something in a protein in the blood, possibly. Not really sure. 25 kb long, and the messenger RNA is only 2.1 kb. And there are 14 introns. Wow, so huge, significant amount of RNAs being transcribed into the pre-messenger RNA and then being removed and disposed of to make the messenger RNA. So it seems pretty wasteful. So we have another one in the table, which is truly, I think, remarkable. Dystrophin, the dystrophin gene, which I believe is the longest gene in humans, which is 2,000 kilobases, so 2 million bases long. 2,000 kilobases is 2 million bases. And the messenger RNA is relatively small or short. It's only 17 kb. So I think if you do the math correctly here, that's 1,983 kb of RNA is removed from the pre-messenger RNA to make the messenger RNA, and this is just disposed of. So as I said before, how many introns are 79 introns. So the important concept here is, even though this is being disposed of, not all of it is junk. There are some sequences in here that are useful. We just don't know why there's so much of it. We don't know why. We don't understand why some genes have such long introns. So if you can figure that out, well, you'd be pretty famous. So, okay, let's talk about splicing. Just a little bit about splicing if you want to talk about the mechanism. If you want to learn more about the details of how splicing works, definitely take BSc 350. We spend a lot of time talking about this complex called the spliceosome. And for this course, you need to know what the spliceosome is. You need to know it exists. You need to know it has about 80 protein subunits and small nuclear RNAs. So the ribosomes have ribosomal RNAs that form part of the ribosomal structure. Well, the spliceosome has small nuclear RNAs. Small nuclear, I got a sharp pencil here. Eventually, I think this tip is going to break. Let's see how long it takes. Small nuclear RNAs. And these are transcribed by RNA polymerase II. So RNA polymerase II transcribes messenger RNAs, or genes that encode messenger RNAs. It also transcribes small nuclear RNAs, or genes that encode small nuclear RNAs. And there are approximately 10 types of small nuclear RNAs in the human genome. So spliceosome, huge complex of protein and structural RNAs that is in charge of removing introns from pre-messenger RNAs. So let me diagram just one pre-messenger RNA, and I will put the 5' cap in here. And I believe the 5' cap is actually added shortly after transcription starts. And I'll diagram the poly-A tail in here, even though the poly-A tail can't be added until the transcript has been cleaved or released from RNA polymerase during transcription or after transcription. So this is our intron right here. And splicing, you don't need to remember this because I need to look it up to make sure, but I believe splicing happens as the messenger RNA is being transcribed, as the pre-messenger RNA is being transcribed. So what we do need to remember is that there are consensus sequences for introns. And we want to remember that the 5' end of the intron, at the 5' end, there exists a 5' consensus sequence for introns. And at the 3' end, there exists a 3' consensus sequence for introns. So the 5' consensus sequence for introns is GU, and the 3' is AG. So most introns start with a GU and end with an AG. And what the spliceosome will do, it will remove this intron, remove the GU, remove the AG, and all the bases in between, and join this exon to that exon. So this would be the 5' splice site, and this would be the 3' splice site. And the 5' splice site is immediately followed by the GU, the very beginning of the intron, and the 3' splice site is immediately preceded by the AG. So let's say the first base before the GU, let's say that's an A. Let's say the last base, the next base after the AG, let's say that's a U. Well, after this intron is removed, the exon will look like this. Well, the messenger RNA will look like this. So that A will be stuck to this AU right here. And we will have our mature RNA molecule. Messenger RNA. And this is ready to be exported out of the nucleus. And the 7-methyl guanine cap, the poly-A tail, these play important roles in proteins binding to the messenger RNA and exporting it out of the nucleus. So before I go on, I just want to address one other thing. So not every GU and not every AG in a pre-messenger RNA serves as the beginning of an intron and the end of an intron. There are other sequences around these that help the spliceosome determine where the 5' splice site and where the 3' splice site is. We still don't know the exact details of how this process works. I'm going to show you an example of a human disease correlated to, or genetic disease correlated to, a mutation that causes a switch in the 5' splice site. We don't quite know why that mutation causes that switch because we haven't quite worked out all the details of what determines a 5' splice site and 3' splice site. Again, we'll learn some of the details that we know in this course right here, BSc 350. So before we talk about that study, I'll do that in the next video, and also in the next video, we'll talk about some of the benefits of introns, some of the reasons why they exist, or the reasons we think why we think they exist, because they appear to provide eukaryotes with some benefits. So, but before we go on, just one thing about the structure of a mature messenger RNA. Structure of mature messenger RNA, or it's just structure of a messenger RNA. So if this is the 7-methyl guanine cap, and this is the poly-A tail, and this site right here, this matches the +1, right, of the transcription start site. And this last base before the poly-A tail matches the transcription cleavage site. An important concept I want you to remember is that this isn't where the ribosome starts translating the protein. That's going to be somewhere down here. So we are going to call this AUG here, the translation start. And somewhere down here, not at the very end, somewhere down here, this will be where the stop codon is. This is the translation stop. Now, the region between where translation starts in the beginning of the messenger RNA, this is called the 5' UTR. And UTR stands for untranslated region. In the past, I've asked exam questions where does this stand for untranslated region or untranscribed region? It's definitely not untranscribed region, right? Because it's already been transcribed. It's the untranslated region because ribosomes, well, in eukaryotes, they bind and they scan for the AUG, but in prokaryotes, they bind somewhere around in the 5' UTR over the AUG. But the important thing is, this is not translated. The ribosome starts translating at this start codon here. So the 5' UTR, 5' untranslated region. And you can probably guess, since the stop codon occurs way up here and the end of the messenger RNA is found down here, that this is called the 3' UTR, for 3' untranslated region. Now the sequences between the AUG and the stop codon, these are the coding sequences. So an important concept here, for a messenger RNA codes for proteins. Does every base in a messenger RNA code for the protein? No, it doesn't. So right, poly-A tail doesn't, right? The cap doesn't. 5' UTR doesn't. 3' UTR doesn't as well. So these sequences are all have regulatory roles. And they're all important for translation, but they don't actually have codons that are decoded by the ribosomes. Okay, so that's it for this video. In the next video, we will take a look at some of the benefits that introns provide eukaryotes, and that concept is going to be alternate splicing. We will also take a look at a genetic disorder that appears to be related to improperly splicing of a gene in humans, and that gene is going to be improper splicing of a messenger RNA from a gene in humans, and that's going to be pol3RA, I believe, which is a subunit of RNA polymerase 3. Okay, see you in the next video.